Hi, this is Dr. Jabali. In this video, we're going to focus on past IB exam papers related to topic 9.4, which is all about plant reproduction. So I went ahead and picked up the most common questions related to plant reproductions, which talks about flowering and seed dispersal and germination. So right here we have the word explain, which that means talk as much details as you can and the whys behind it. Let's see here. Explain how some plant species are able to respond to changes in their abiotic environment and flower at the precise time of the year. So from this, the first thing it says we're talking about plants. Okay. And then it says respond to changes in the abiotic environment. When you talk about abiotic environment, we're talking about changes such as maybe the temperature, maybe the sunlight, maybe the amount of CO2, whatever it is, humidity. So this is what abiotic we're talking about. And flower at its precise time of the year. Now, when we say able to respond to changes, basically we're talking about the stimulants here changing the shape of the plant. And explain here the flower at the precise time of the year. So that means we're talking about flowering. Now, when we talk about flowering, we definitely are talking about the phytochromes, right? That's one of them. And when you talk about responding to changes, we also are talking about changing the shape of the plant, which is going in a certain direction, it's increasing, like for the sun. So this probably talking also about the shoot apex and how you have the meristems eventually creating, for example, more stem, more leaves, right? Oops, wrote the leaves wrong. More leaves, or even roots, whatever it is, it's causing it. Of course, that would have been the shoot apex, and all of this is obviously working with the auxin hormone. If you remember the auxin hormone, which grows the shoot apex. Now that would have been if I was talking about growing basically the stem, but when it comes to the flower. In this case, it will talk about the phytochromes. We talk about the PR and the PFR and how you have short days that want PR and the long days that want PFR in order to flower. And this is all based on the night. The more night exposure, the less night will tell you if you're going to have more PR or PFR and eventually go through the process. So let's look at the mark scheme. Genes for the flowering are activated, gene activation, gene expression. So basically, yes, when we talk about the genes, there's a gene that will be activated and it will allow the flower to happen. And once this gene activated, then eventually you're going to have the pigment. The shoot apex changes from producing leaves, stems to producing flowers. So if you remember the shoot apex, which was right here, right? This shoot apex, I was just telling, they have the meristem. So if you decided it's time to flower, then instead of growing the plant this way or growing a leaf, what does it do? It turns this off and it turns into breaking this into make it a flower. Now, day length, duration of day, night length, part of peer measure, detector, responded to. So again, like I told you, it's all based on the day and the night length in order to see if it's flowering. Short day flowers flower when they have a long night period of darkness, and long day plants only flower when they have a short night period of darkness, which basically I was talking about the PR and PFR. So the short day plants flower in the late summer, fall, autumn, and the long day plants flower in the early spring to the early summer. So this is basically, again, like I told you, the short plant, right? It wants a lot of PR. And if it wants a lot of PR, that means it needs a long night to turn all the PFR back to PR, while the long day plants, these guys want a lot of PFR, and that means they want short night. So there is very much less conversion of PR into PFR. You don't want much of that. That's why it's a short night. So there's only maximum this, so it doesn't even ask about it doesn't even has the mark scheme talk about PFR and PR, but it does here mention about short days. They have to do with the long nights, or the long days you have to do with having short nights. You also talk about the short days. They will flower in the late summer, while the long days will do mostly in the early summer. Now, besides that, we also talk about the shoot apex, right? Because they usually produce leaves and stem, but because the gene expression now changed to flowering, it's activated. Now we're going to do flowers instead. But again, it's all based on day length and night, or photoperiod, which basically is detecting the light, and from there the phytochromes go. So in this mark scheme, you don't see anything talk about phytochrome. It's very general. It talks about short and long days, and then it talks about gene expression. So I would add this in here because you don't know in the future mark scheme it goes more in detail. Now the next question says explain how flowering is controlled in long day and short day plants. This is again, we're going to talk about the long day. The long day, they usually have this phytochrome. Now the phytochrome is basically a pigment, right? And if it is active, this pigment, then it's called PFR. And if it's inactive, then it's PR. During the day, PR is converted to PFR. And during the night, PFR is converted to PR. Now this is done very fast and this is very slow. So long day, for instance, he wants what? He wants a lot of PFR. That's why during the day you have a lot of PFR produced. And short day means that you want to have a long night, because in the long night, what do you want to produce? You want to get every PFR back to PR because PR is what you want. So this is how you're controlling this, and you can manipulate this in the greenhouses. So you can do this off season. So let's see. Flowering is affected by the light. See, the most obvious things, I did not talk about this, but it's the light. Phytochrome, we did mention, is in two ways, PFR and PR. PR converted to PFR during the daylight, and sunlight contains more red than far red, so the PFR predominates during the day. So here I have an additional thing that I can talk about is the red and far red light, which obviously the red on the 6 is the PR and the far red is the PFR. So this is an additional information that I did not mention, but now it's here in the mark scheme. The gradual conversion of PFR to PR occurs in the darkness, as I mentioned. The PFR is active form, PFR is active, PFR is inactive form. A long day plays are induced by dark periods shorter than the critical length, occurs when the day is longer than the critical length. And enough PR remains in the long day plants at the end of short nights to stimulate flowers. So basically just saying that the long days wants the PFR to flower. So you get the points. PFR acts as a promoter of the flower in the long day plants. And then short days induce flower by dark period longer than the critical. So that means we definitely want to have a long period of night. So the day is shorter than the critical value. And the long nights, enough PFR is being converted to PR. So that means the flowering will occur. PFR acts as inhibitor of flowering in short days. So obviously, PR causes the flowering while the PFR will inhibit it. And the same thing goes with the long days. PFR will cause the thing while the PR will inhibit it. So looking back here on the mark scheme, the things that I mentioned will get me the points. The phytochrome, they exist in two forms. The PR is converted to PFR during the day. Gradual conversion of PFR occurs in the darkness. PFR and PR, the active and inactive form. So the long day induced by dark periods shorter than the critical length. PFR remains in the long day at the end of the short night, stimulate flowering. So the PFR acts as a promoter. That means it's pushing the long days to flower. Then, of course, the short days, the induced by flower, are dark periods longer than the critical length. That means the days are shorter than the critical value. And the end of the long night, enough PR has been converted to PR, so a lot of flowering. So PFR inhibits this one I did not mention, so I'm going to say this one now. But I didn't mention most of it. So now that I know the mark scheme, I have additional ideas that I can talk about. For example, the obvious thing is saying that it's affected by the light. And then I talk about the red and the far red lights, which I did not talk. And talk about here the PFR acts as an inhibitor for the short day plants. The rest is, of course, mention the critical length, mention if they want more day or they want more night, mention about PR and the PFR, mention about how they convert into the day and who's converting during the night, what does the short day want, what does the long day want. Mention about the phytochrome, there are two ways, which is active and inactive, If you have this all here, then you have a strong base mark scheme to any time you want to talk about these two. So the next question says, in an experiment was done to test a hypothesis that temperature affects the rate of germination in broad things. So we talk about how temperature will affect germination. Now outline, so of course this bullet points, 2 factors. So we want 2 factors right here, apart from temperature. Okay, so we do not want to mention temperature. That should be controlled in the experiment. So when we think of germination, we know that temperature affects it, we know that oxygen affects it, and we know that water affects. These are the main things needed for germination. This is no information. So the 2 other factors, we definitely would say that it's oxygen and water. These 2 can affect the germination of the experiment. So let's see the mark scheme. Same amount, type of soil, substrate, pH of the soil. So this is one example. Another one is the same amount of water. So you can see that the water, I have mentioned water is one of the samples. And oxygen. So these 2 I have mentioned here that does affect germination. So I got the 2 points. But now I also know that I can also talk about the type of amount of soil. So they can talk about the pH of the soil. So the soil does affect the germination too. So now I have an extended information for this. Same measurement of germination and time. So these 2 I have not mentioned, but I did mention the amount of water and oxygen. Because again, temperature, oxygen, water, these are 3 things that is necessarily for germination to occur. But now the mark schemes additional information such as the time of germination and the amount of soil. Now I have expanded my mark schemes, so are you. So whenever you have a next time, make sure you put those. But one of the most common answers is definitely water and oxygen and temperature. So keep that in mind. And then here, same number or source of agent seeds. So these are another ones that can actually cause as a factor. So this is like basically doing an experiment here. So once again, these guys here are additional things, because we're talking about making a test of temperature affecting germination. And that means if I want to test germination, what are other ways that I can test germination? Why not playing with the soil? Why not playing with the time of germination? Why not talking about different seeds with different ages? Or the numbers of seeds? So these are just ways to manipulate different experiments with germination. Accept discussion of light only in as much as related to temperature. So basically, temperature, light are not the same thing, but it's if you discuss more about the sunlight, not necessarily temperature, then it's okay. So all of these here are just ways that you can manipulate an experiment doing with germination. The next question says here, describe the metabolic events of germination in starchy seeds. So all of this starts with water, which is basically imbibition. So that means the seed absorbs water. Now, when it does this, then the seed coat will rupture. At the same time, the metabolic process starts. So then it releases gibberellins. And gibberellins, what it does is a hormone. And this hormone, what it does is activate an enzyme. Now, the name of the enzyme in this case is amylase. And what does amylase do? It basically breaks starch into maltose that eventually will be broken down to glucose. And of course, cell respiration will happen, which you're basically going to use glucose for the energy. And there's going to be a lot of motion of the sugar for the metabolic rate of basically the seeds turning into a plant. So this is basically the things that's going to happen every time we talk about the metabolic events of germination in starchy seeds. So let's see what we have here for the mark scheme. Absorption of water, and then increased respiration, which of course I told you about cell respiration happening. Secretes gibberellins, and then secretes gibberellins what? To stimulate the production of amylase. Digestion of starch to smaller sugars, maltose. And then here, mobilize tissues, transport food, nutrients to the embryo. So of course, this is the moment I'm talking about the metabolic reactions in which you're using glucose for energy. And that's pretty much it. This is for this question. So you can see here, if I target the points, we did this, we did talk about separation, we talk about turbulence, about MLAs, and this. So you probably got all the points because the mark scheme will never say you have to answer all of these. So you fully got the points. So once we look at metabolic events of germination, you have to mention about the water. You definitely have to mention about the hormone gibberellins, the enzyme MLAs, and what does he break it down. These are the most common things that comes when talking about germination. And I'm going to be careful between metabolic events of germination and the factors that affect germination because they're completely different. This is talking about the steps of germination once you want to open the seed. And when we talk about factors, then we talk about the temperature, the oxygen, and the water. These are the things necessary for germination to occur. But once germination occur, these are the things that happens once germination is occurring. So the next question says here, draw and label the testa and the radicle. So let me go straight to the drawing here that it already shows. So you can see that this is basically the drawing of a seed, and you can have the testa, which is about here, and the radicle, which is on the inside. So again, you can just do a drawing like this, and you can put this as testa, and then you have something small like this, and you can say the radicle. So make sure to label them and draw them, and you get the full points. Next, draw a half view of an animated flowers. Again, you can start with here the stigma with the style, and then the ovary. So you can have here the stigma, the style. This is the ovary. Make sure you label them well. And then you can have here the ovule if you want. And then you have a line coming out here, and you can say that this is the filament. And this is the anthem. If you want, you can say that this is the stamen, which is the part of the man. And you can say that this is the carpel, which is the female. And then after this, you can do a round like this and have this as the petal. If you want, you can draw this the sepal. But this is plenty of enough. Make sure to label them well. Make sure to put them on the right spot, the line, as well as the word. So let's look at the mark scheme. So here is the picture you can see, right? So you don't have to target all of them, but mostly target the anther, the filament, which makes stamens, the stigma, the style, and the ovary, which makes the pistil and carpel the same thing. You can also have the petal right here, and the sepal and the ovule. You don't need to know the receptacle and the pentacle on this, but these ones are plenty of points. And you can see it's usually 4 points. So we got the sepal, which is the outermost part of the flower. We got the petals, which is the part, the largest part of the flower. You don't have to write. Basically, it's just a drawing. The stamen, and you have the anther and the filament in there. You have the carpel or the pistil, which is the stigma, the style, and the ovary, which you have underlined here. The underlines are the ones that has to be labeled. Now, you can have the nectar in the base of the ovary if you want, and the ovule inside the ovary. So we did get more than 4 points here, but the ones that are underlined are the ones that have to be labeled. Now, here says, as the question does not specify a labeled half view, allow some marks for unlabeled structures. Award 1 mark for any of the 2 of the 6 structures in the mark scheme. MPA to MPF. It must be clear what each unlabeled part is. The maximum mark is therefore 3 for an unlabeled mark half view. So basically, we have labeled all the points here that we can think of. So you get a full points for this. Next, we have here, it says, successful sex reproduction in flowering plants depends on several essential processes. Outline the role of pollination and seed dispersal. So we're talking about pollination and seed dispersal. Now look at the next question. The flower of angiospermophyta are used in sexual reproduction. See, it's still talking about sexual reproduction and flowering. Outline 3 processes required for the successful reproduction of angiospermophyta. Now, one of them, it directly goes straight to the point and tells you pollination, seed dispersal. But the other one, it does not mention them. Now, here's the most important thing to see how those 2 questions are exactly the same. It says successful sex reproduction. So you can see successful reproduction. When I say flowering plants, it's the same thing as angiospermophyta. And then it says several essential processes. So it says 3 processes. Now here it says flower of angiospermophyta, which basically it's, again, the flowering and sexual reproduction. So you can see they're all talking about sexual reproductions of flowering, and there's a central process. On the first one, it gives away two of them, pollination and seed dispersal. And the bottom one, it talks about three processes. So they're both still talking about basically the angiosperm phyta, the sexual reproduction. So when we say pollination here, we're basically talking about pollen going from the anthem into the stigma, eventually to the style, right? And from the style, eventually to the ovary to get to the ovule so you can do fertilization. We got seed dispersal. And this is where after fertilization, you're going to have the fruit being grown, and then the fruit can be eaten by other types of animals, and these animals can just fly these seeds of the fruits and throw somewhere out. So it's basically the seeds of the fruit that is thrown out somewhere. So that's what seed dispersal is. The pollination, for example, pollen can be carried by bee or by insects, right? Which they touch the flower because of the scent of the flower or the color of the petal, and they touch the pollen and then they fly it. Or it can also be by the wind, which the wind can fly the pollens into the stigma. So these three guys can just take the pollen into the stigma. It can be in the same flower or it can be to another flower. So if it's the same flower, we call it self-pollination, right? And if it's different flowers, we call it cross-pollination. So let's look at the mark schemes here. Now you can see the two questions are asking the same thing, but let's look at their mark schemes difference. Pollination, transfer dispersal of pollen from the anther stigma to the stigma. See? Or transfer dispersal of movement of pollen between plants, allowing fertilization to happen. Seed dispersal, distribution of seeds so the new plants have space, nutrients to develop, avoid competition, colonized habits. So it's just basically you saying the distribution of seeds. Accept any other valid role, accept to other words of fact. Answer must be about the role. So once again, seed dispersal is just throwing the seeds to another area, to maybe an area that has most nutrients. Pollination is getting the pollen out of the anther, into the stigma, all the way to the ovary. The bottom here, what we have, the three processes required for sexual reproduction. So one of them we actually have, like the top is the pollination, transfer pollen from the anther to stigma. We also talk about seed dispersal, which is spread of the seeds. Now these two are mentioned on the top. What is not mentioned on the top that can also be is like the fertilization that happens after pollination, which is the joining of the gametes. And there's also meiosis, which is the production of female and male gametes, right? When you're producing the pollen, the sperm, and the ovules and the ovary. These are meiosis. Now it says reject fruit dispersal because it's not fruit that you are dispersing, it's the seed that you're doing. So in this question, it outly asks you for only for 2, and this one asks for 3, but they're still talking about the whole sexual production. So these two basically are like these two, but we also have now here meiosis and fertilizations, which these are other sexual things that is happening. And of course, meiosis to produce the sperm and egg, and fertilization is the join of the sperm and egg together. So these are pretty much the kind of questions that comes from 9.4. Now here are future questions that can come from 9.4 that hasn't been asked yet, but these questions were mentioned in my lecture, so it can help you. So just make sure you know very well about the flowering, its structures, the short and the long days, the phytochromes. Learn also about seed dispersions, fertilizations, pollinations, as well as the germination. These are the most common things that can come out from 9.4. And that's pretty much it. Thank you.